everyone, myself Dipanshu Demi, and I am a student of Master Degree final year of Vishwavarthi University. Today, the topic I am going to teach, that is the sliding filament theory. So, what is the sliding filament theory? Basically, we know that to move our body from one place to another place, we generally we take help from our voluntary muscle, that is our skeletal muscle. So the process that happened inside the cells and inside our muscle fibers, that is called the silent sliding filament theory. Okay, so let's start. Okay, so before that we must know that what are the components are used in sliding filament theory. Okay, so the things that are being used, that is the actin and myosin. So this is the actin and this is the and this is the myosin. So we must know that what are the component and what are the areas called as different. The areas are called differently and they have some different names. So we must know also those. Okay. So you can see there is a tail after the myosin head. This is called the myosin head and this is called the tail. So the tail part is called light meromycin. Okay, you can also say light. You can call that light meromycin. LMM, and the head part is called heavy meromycin. That is HM. Okay, and the cross between two mycin is called cross arm. Okay, you can see the cross here is called a cross arm. And in the myosin head, some things are already present. That is, one thing is a actin binding site ABS, and another one is this is also called ABS, but it is this is ATP binding site. Sorry, I made a mistake. This is not ABS. Oh, this is ABS. Sorry. So this one is also ABS, and this one is also called ABS. This one is two things are present, that is the actin binding site, and another one is ATP binding site. Okay, clear? I think. Let me remove this. Okay, so this was the structure of myosin. Now what is the structure of actin? So you can see there is a yellow line and some bubbles like shape is there and some three dot. This is a complex structure. So if you see, if I draw a easier structure, so the structure of the actin will look like this. This is the bubble like structure, as you can see. Forgive me because I'm using my mouse for drawing. That's why the drawing will not that good. Okay. So just imagine that this kind of bubbles are present also in this side. And there is a line of protein is also present between this. Like this, the here it is yellow. I draw it in the red color. And as you can see, there is three proteins are there. Let's draw those on also. Three protein are present in the cross of the, you can see the cross, there is the protein is present. In somewhere the protein is present here. it's that all have some specific names. Like these three proteins are also called TM. That means it is mean trimeric protein. These three proteins are also called trimeric protein. It is made of three protein. That is, if the protein is look like this, so one is called TPC, another one is TPI, and the last one is TPT. Okay, you can see that it is been written in the image also. Okay, so it is made of three protein, that's why it is called trimeric protein. And the, the bubble like shapes are called globules, and the yellow line that is called the tropomyosin. You can see this is troponin. Three proteins are called troponin. Troponin is also called the trimeric protein, that is made of three protein, that are TPC, TPI, and TPT. Okay, this is the whole structure is called as actin. So I think you have understood the structure. Okay, now there is some condition for muscle contraction. Okay, so as I told you that it is the myosin head. In myosin head, there is actin was present. Actin was present in the head of myosin, and there is a protein is called trimeric protein or troponin is present. The conditions are until in the head of the myosin, actin is present in the head of the myosin, the muscle contraction can't be happening. And the condition in case of actin, that is, if the trimeric protein do not shift their place, like change their place from this place to a certain amount of travel a certain amount of area, the muscle contraction would not be happening. Okay, so for that, in case of myosin, to break down the ATP in the head of in the head of myosin, a activity or a activity, we can say a activity is present, that is the ATPase activity. This is a kind of enzyme that helps to break down the ATP into ADP and inorganic phosphate, that is called also PI. And to remove the troponin from the actin, or shift the troponin from the actin, it is a long process that we are going to start now. Okay, I think from this part is clear from your side. Okay, so now we are going to start the system that is called the TTBU system. Okay, so this is a what is this called? This is called a neuron. Okay, and this is the action, and that is the action terminal. Okay, so in action terminal, some neurotransmitters are present. Okay, so when the signal from brain comes to our neurons, uh, it ends in the action terminal and in the muscle fiber. Okay, when the signal is came, it releases some neurotransmitter, where in this case the neurotransmitters are acetylcholine. Okay, so when the acetylcholine is entered in the muscle cell, now entering the muscle cell, it attaches to the receptors of cell membrane or plasma membrane. Okay, through plasma membrane, uh, it enters the muscle fiber, and now it when the muscle fiber it is inside the muscle fiber, it depolarize the muscle fiber, and also you can say the plasma membrane, it depolarize the plasma membrane, and the plasma membrane releases sodium from it. Okay, now the sodium is being released. Now the sodium reaches to sarcoplasmic reticulum. What is the sarcoplasmic reticulum? Now sodium is being released from here. Sodium will come and enter in the sarcoplasmic reticulum. This is the sarcoplasmic reticulum. It enters the sarcoplasmic reticulum, and the body will get shocked to see this much of sodium. So in that by the reaction of this, the sodium the sodium will lead to release calcium from the sarcoplasmic reticulum. Okay, so now the sarcoplasmic reticulum has released calcium, and then now the calcium, what the calcium will do? We've already known previously there was a protein in the trimeric protein that was called TPC. It is which is made of sodium. Okay, that is made of sorry that is made of calcium. That is made of calcium ion. Okay, so now the sodium released by the sarcoplasmic reticulum will reach to the TPC, and the TPC will now, if the protein enjoy it, if the protein, you know that protein is present in this area only. If the protein was present in this area, after the reaction between the calcium and the TPC, it will lead to move the troponin protein from this place to slightly away. What will happen for that? This will open the myosin binding site. So there was the myosin binding site which was covered by the troponin protein, and now after the reaction between TPC and calcium, the myosin binding site is totally open, and it is ready for the myosin to attach with the myosin binding site. And now the myosin is ready, because we already know that because of the ATPase activity, the ATP is removed, and the ATP is break down into ADP and PI, and also after the reaction between the TPC and Ca plus, that is the sodium, the myosin binding site is also opened. Okay, so now next, what happened next? Let's Okay, now the both conditions are fulfilled. We will start from here. We start from here. So first, their ATP was present in the head of the myosin. So no contraction was able to happen, because there is some certain condition that if the ATP is present in the head of the myosin, the muscle contraction will not happen. Now you can see the ATP. Now in the second image, the ATP is breakdown into ADP and P. That will be PI, that is the inorganic phosphate, and ADP you already know. How this is happening? Because of ATPase activity. Now the ADP is totally ADP and PI is ready, and also the actin is also ready. Now when the myosin binding site is open, the myosin is immediately attached to the myosin binding site of the actin. You see the myosin head is attached. And lastly, when the when the ADP and PI releases, ADP and PI was present in the head of the myosin. When both of these was released from the head of the myosin, the certain stroke is happening. Okay, now see, first the head was like this, and in this image, the head is moved from here to here. Okay, from here to here, from here to here, the head is being moved, and the muscle contraction happen this way. You have to remember that myosin never moves from its position. Okay, and only the head of the myosin is moved, and it forces actin to move from one place to another. Now the ADP is released from the head of the myosin, and again if there is nothing in the head of the myosin, the ATP will automatically come in the head of the myosin, and this process will continue for long until you doing the activity. Okay, now see some videos. Watch this. The calcium ions are coming, and there's a troponin. Calcium ions are coming, and now it shifts the actin binding site. Now the actin binding site is open. Okay, now the actin binding site is open. Now the myosin the myosin head can come and attach to the actin. Okay, what is the next step? There is the cross bridge. Now you can see there is myosin. The myosin will come. The next step, see the power stroke. Now this was our ATP that is removed, and now after the ATP is removed, the myosin head was free, and now it attached and stroke by the myosin. You can see that myosin did not move from its position, only the actin is moved. Okay, so let's see the last step, reactivation of myosin head. Now the contraction has been done. Okay, the ADP and PI is gone from the myosin head, and ATP again restored. ATP's activity also restored, and the myosin head came back to its natural or its initial position. So if you see this structure, this is called the sarcomere, and in sarcomere this is the myosin, and that is called the thick filament, and this is the actin. And this is the state of a relaxed. This is the state when the muscles are in relaxed position. You can see this written that relaxed muscle, and this is the state when the muscle is contracted. You can see the length of the actin is decreased when there is the muscle contraction, and when the muscle is relaxed, the the length of the actin is longer. But you can see that the length of the myosin in relaxed position or in contraction position, the length of the myosin is same. Okay, so this is one of the characteristics of sarcomere. Let's see. So now there is some question, previous year question that already came in UGC net, that I have given here. The common question in NEET, that is the sequential question. The examiner will ask that what is the sequence of sliding filament theory. This type of question generally asks. So there is another question that is this is also a sequential question. The option will come like this also. So this is the another question. And this is the last question. So thank you everyone. I think I have helped you on this topic because there was some limitation that I was doing the presentation through a mouse. That's why I think that explanation was not that level on that level. So I hope still I able to give you some extra knowledge. Okay. Thank you everyone. That's all.